Hello, everyone. Welcome to the seminar. Today, I'll be introducing Dr. Abbie Smith. She's an assistant professor of medicine in the Division of Infectious Disease, and she's also the director of combined laboratories of the Emory HIV/AIDS Clinical Trials Unit, Infectious Disease Clinical Research Lab, and the CFA. Dr. Smith's work with the Translational Mucosal Immunology team at the Emory Hope Clinic studies HIV and SLV evolution, transmission, vaccination, and functional cure within human and non-human primate models. Her group places special emphasis on sexual and gender minorities as well as other marginalized groups. Through understanding how sex and gender impact gut mucosal immunology, HIV pathogenesis, and product formation, their research can inform novel, personalized HIV prevention and treatment strategies. So I just want to welcome Dr. Abbie Smith.
Thank you very much for the invitation. I'm really excited to give this talk today because I didn't know this training grant existed and like this is what I do. Like since I was in graduate school, I clicked around to get more information about this group and I saw the training team. There's something here on landscape genetics. Every single paper I've written has the word landscape in it. But like I love it. I love thinking about sequence space and viral fitness landscapes and antibodies landscape. Like that's what I do. I'm so excited. I'm going to walk you through some of the research I've done over the years, starting with HIV population dynamics, which is quite different than, let's say, E. coli or blue whales. It's quite unique, and I want to walk you through some of those details. But then ultimately, my research evolved into also encompassing the B cell evolution, evolution of the virus, and then taking into account like getting even bigger across the scales. HIV and its interactions with our immune ecosystem, not only B cells, and the research we're doing today on how all of these interactions influence disease outcomes. And I've got this nice diagram here, but my lab might describe me more like this. So some of these concepts I haven't gotten to talk about in a very long time, but there's something I'm really passionate about them. So I'm a little rusty, thank you for your patience on some of this, but also I'm really excited. So to start out, I heard you all wanted to learn a little bit about just my background and also professional development. So I'm from a small town in the middle of nowhere in Missouri. We are known for Jim the Wonder Dog, who is a psychic dog. Basically what happened was during the Depression, a gentleman was trying to figure out how to keep his family fed, and he worked out this scam with the psychic dog. And we're very proud of that fact. But that's another way to say, where I grew up, I thought if you were good at school, either became an engineer or a physician. My brother's an engineer. I had a panic attack in a 3M factory, so I thought I'd become a physician. I'm a little bit awkward interacting with people, as you might pick up on. So I knew I wasn't going to become like a nice family practitioner. What I wanted to do was study criminals. So I studied my undergrad under this individual, Sal Costa. This was before like true crime podcasts and law tubes and that sort of thing. He studies psychopaths and serial killers, and I researched with him. And it was my plan to become a psychiatrist and doing that kind of research. I would have bet you $1 billion. That's what I was going to do when I grew up. And this is my favorite serial killer. Like I'll swing up. Look him up, we learn a lot about how he was able to get away with his crimes for so long. But what happened was I was doing all this research and I applied to the FBI's summer undergraduate program. And I don't know whether they just never responded to me. Like I had to type that application. There was no online portal at that time. And so I never heard back. So I applied to other summer undergraduate research programs and was given the opportunity to do so by this gentleman here, Charlie Wood at the Nebraska Center for Virology. I had no idea what I was doing. I thought it looked great on med school applications and doing HIV research in his lab under two investigators from Zambia, Solid and I. They taught me, like they gave me my training wheels and let me fly with academic research science. I didn't even know that this career was a possibility. But through that program, I had so much fun. and learn new things. That ultimately I did my PhD program with Charlie's protege, John West. They're actually both at LSU right now. And so what I studied is HIV population dynamics. So if you all haven't heard of this person, Manfred, you can Google it. He has passed away, but I think if he were a productive research scientist during modern times, he would be having so much fun. He's crazy in a good way. But he came up with this idea as a chemist of sequence space. So think of a cube. In that cube is all of the potential sequences that a species is currently exploring. So let's say your humans are this sphere in the lower left. We're exploring some sequence space, but for the most part, humans are quite similar to one another. But compare that to this tiny dot. Think of like a monoculture of like a GMO papaya. It's basically a speck of space of papayas. Viral populations, specifically RNA viruses, operate quite differently. And so what Manfred suggested is that RNA viruses are more like a diffuse gas within this cube that's constantly like shifting and moving around. And so this gas might have a consensus sequence, but if you selected any individual virus out of that cloud, it deviates slightly from the consensus. And so this is called a quasi-species. And just to really emphasize how specific HIV is compared to even other RNA viruses, this is a illustration from many decades ago, but I think it illustrates it nicely. That in the upper left here, we have a phylogenetic tree based on genetic diversity within the 1996 influenza season. It's comparable to the HIV sequence diversity in one person in several years. And then you compare that to the diversity observed in an entire cohort versus all of the sequences isolated from the Republic of Congo in 1 year. The kinds of diversity you see with HIV is really quite incredible. So how that happens for those of you who are familiar with HIV. So here's the virus, and there's a protein on the outside called envelope, and that is the only target for neutralizing antibodies. It binds to the host CD4 cell, co-receptor, mediates infection. And then what happens is that RNA genome is reverse transcribed into DNA and then permanently integrated into the host cell. Then it's treated just like any other host gene, transcribed, translated, and assembled in a progeny variants. But where we get the diversity, you might know, is from reverse transcription. It doesn't have a proofreading step to it, so we get mutations there. But also, HIV is functionally a diploid organism. There are 2 copies of the genome per virus. So as reverse transcription is happening, it can hop back and forth between those genomes. And that's how we get insertions, deletions, we get entire gene duplication and then subsequent divergent effects. And then also, let's say 2 totally different viruses infect a cell. 2 different genomes can get packaged in this progeny virus. So the next time it goes to infect a cell, it can hop between 2 totally different viruses and get circulated more commonly from the cell. So I want to highlight here 2 things. One is that all of this sequence diversity is not happening just willy-nilly. This is evolutionarily defined diversity. If it mutated too much, then you would introduce all of these deleterious mutations to extinction. You would have to have an immune response and burn itself out. Furthermore, this diversity builds itself, rebuilds itself, recreates itself over and over and over and over, every single transmission event. So this is just an illustration showing lots of viral diversity you see during chronic infection. But when you look in the genital tract, there's less diversity. And then what's actually in the genital fluid, even less diversity. What makes it into the recipient is even less. What actually establishes new infection, new person, is almost always one genetic variant out of all of the diversity. So one genetic variant, imagine if you had a population of penguins and every 5 years you reduce that population down to 2 penguins. Ultimately, just due to chance, founder effects, it would lead to extinction of that population of penguins. This happens tens of millions of times, and HIV has not gone to extinction. It is just part of the nuts and bolts of how this virus works. So, when I was in graduate school, I studied a number of individuals, but this one represents a very famous individual, 1157I. So this infant was infected with HIV in utero. So normally that is, it's always a death sentence. But being infected in utero as opposed to being infected during birth, generally has much more aggressive disease progression, especially during this time when access to antiretrovirals was more limited, especially in this infant grew up. And so this provided us with an opportunity to sequence the virus in this young person to see how it changed over time. And so specifically here, the evolution of the gene that goes outside. And so I just want to highlight a few things. This virus that was isolated 0-month time point is a subset of all the diversity solid mother and transmission. But as time goes on, you see, for example, let's say 67 months after birth, there's a lot of diversity, but there's little flashbacks of sequences that are more similar to, let's say, 24-month or the 12-month of these kind of re-emerging latent viruses that are just sort of testing the water again. Is it safe to come out? Am I needed? No, okay, I'm neutralized. And so this diversity is the end result of selection based on the patient, the infant's antibody response. And so we see the 6-month virus here is readily neutralized by plasma collected at later time points. And indeed, plasma from the mother could even neutralize this virus, because it's quite similar to the viruses that she was living with. But when we go to 48 months, we see that only the 67-month time point is able to neutralize. So what we're seeing is the virus population changing and the antibody response being reactive, chasing it, but never catching up. So what's this concept called? It's called the red queen hypothesis. Everything's mutating, changing as fast and fast and fast as they can, but everybody's functionally staying the exact same place. And just again, to go back to professional development, this is a quote from Lave and Valen. I thank the National Science Foundation for regularly rejecting honest grant applications to work on real organisms, thus forcing me into theoretical work. I think we can all feel that. And he established this foundational evolutionary concept as he had to. Yeah, what about my particular? So my question was during grad school. We've got all this diversity, this viral envelope proteins changing. What effect does all of this change have on the fitness of the virus? Because what other protein is comparable in our body that's changing all the time? It's still like hemoglobin's changing all the time, working just fine. What's happening? So again, to go back to some of the foundational concepts, what I have here is a super simplified version of again the fitness landscape, the sequence space. So on the x-axis here are all potential sequences. And canonically, we think that there's a collection of sequences that are most fit. And let's say antibodies are neutralized the virus and you have to move away from that peak, there's going to be a loss in fitness as the population has to move away from this. So that's exactly what happens in response to cytotoxic T-cell responses. That's exactly what happens in the response to antiviral therapy. You might escape that drug, but it comes in fitness cost. What's happening with it? So here's another concept called competitive exclusion. So when you're talking about fitness, that's quite different than replication capacity. You can have two viruses, let's say the red here, green virus, that when they grow in isolation, they're functionally identical, both equally replication competent. However, if you put both of those viruses in the same well and make them compete for limited resources, one of them is going to win. That's just very simplified way to look at that. So I did that with a bunch of viruses. And what I found is that rather than seeing fitness peaks and fitness valleys, I just sort of saw noise about the mean. Well, what does that mean? So this is a different concept. Instead of survival of the fittest, this is survival of the flattest. in the sense that, you know, you can move away from that selective pressure, you might lose a little fitness, but then you move along and it comes back. It's not a big deal, you're not getting trapped in these fitness values that we see, again, with escape from antiviral therapy. So I also characterize a bunch of different steps in the HIV life cycle where envelope plays a role. And it just turns out that some mutations make you a little better at this step, but then it makes you a little worse at another step. And everything just sort of comes out in the wash. So the HIV envelope protein really is demonstrating not only extraordinary genetic plasticity, but also functional plasticity. It is the only target for antibodies. So what does that tell us about vaccines? So in a way to answer that, I did my postdoc at Primate Center with Cindy Gerdine and Eric Hunter and his wife Susan Allen and their cohorts, also Zambia and Rwanda. So my PhD mentor got his PhD with Eric, and Cindy was a postdoc, but he was getting his PhD, and so I became my postdoc. But initially with this project, I was still only studying HIV population over time. But as part of my project, another postdoc was supposed to be looking at B cell, the autologous co-evolution of the B cell response in the patients. And they were dropping like flies. And I wasn't super interested in B cells, but someone had to do that side of the project. So I was like, well, I'll figure it out. Turns out it was a ton of fun. I absolutely loved it. And the overall idea was, you know, we generated lots of vaccine strategies, and this goal of eliciting broadly neutralizing antibodies to vaccine. So one antibody that can neutralize innumerable different HIV variants that an individual could encounter. But we didn't actually know at the time a lot about what was actually occurring in people, because viruses are not evolving in isolation. They're all together kind of swarm. At the same time, B cells are also co-evolving, again, as a swarm. And so again, at the time, there was a new idea, new push towards germline targeting. Yeah.
So I'm curious, how would the introduction of powder in terms of how close the virus needs to be, are the ones that are more extreme, more likely to escape our treatment, or the ones closest to?
So that's it. So the population of viruses as a whole is for the most part going to be susceptible to antivirals. Initially, when we introduced one antiviral, it was easier for those very few to that still had a slight bit of replication capacity to start building up fitness in response. So that's where when we learned that you have to get 3 drugs at the same time, because that just made it harder, because it's just by chance. Right. By chance, there is a variant in every single person living with HIV that it is going to be a variant they're resistant to whatever you throw at them. And then from there, it's just time, not more. But as far as the consensus, for the most part, most viruses are going to be susceptible to these antivirals because they attack very critical enzymes that do not have the same plasticity phenomena. But there's always going to be one. And then it just buying lottery tickets every replication. Eventually. But yeah, as I was saying, there's this push to see whether we can contain the chaos of antibody maturation and force the evolution of a specific B cell they're interested in to generate these broadly neutralizing antibodies. Broadly neutralizing antibody would protect everyone. But these B cells, again, are not evolving in isolation. There are a ton of different B cells in any given lymph node responding to tons of different antigens. So what is going on the macro scale? So to do that, we characterize broadly neutralizing activity in lots of patients from Eric's group, but we saw a broad range from this elite neutralizer at the top to very poor neutralizers at the bottom. And indeed, the reason why we selected one of these R669 for further characterization moving forward is that this individual had very little autologous neutralizing activity. So not just lack of breadth, but neutralizes environments. So my project here was to use each individual's transmitted founder virus to fish out antigen specific B cells in every single person. And I got about 400 functional monoclonal antibodies that were sequenced, cloned, characterized, probably twice as many that just weren't functional for some reason. I don't remember most of this part of my life. I would recommend doing a project like this in the future. But what we found, actually, in here, again, focusing on the elite neutralizer and the poor neutralizer. What you're looking at here is a ribbon plot, and all of the blues are BH1 lineage antibodies, 2 in yellow, 3 in red, 4 in green, and 5 in purple. And then the ribbons are breaking off into different light chain populations. So you can just looking at, you can see, even though there's about the same number of antibodies isolated from both individual, in this elite neutralizer, there's lots of ribbons, lots of thin ribbons here, versus lots of thick ribbons for neutralizer. It turns out of the, about 75 antibodies, there were about 21% that represented components of the same antibody isolated a number of times in the elite neutralizer versus 56% the poor neutralizer. And then when we looked at affinity, the affinity for that autologous protein was much higher than the poor neutralizer, and the number of the clonal families matched out the instrument. They just bound so hard to that protein. And it turns out those antibodies don't have any effector function. So what's happening during B cell affinity maturation is you get random mutations, somatic hypermutation, and the B cells are competing for survival signals. What's happening in that poor neutralizer is he's got B cells that are just fighting so tight and not doing anything. Versus the poor neutralizer has lower affinity, there's more B cells, there's more clones, there's more options. And so when the virus changes, that individual's B cell repertoire can go, oh, okay, we're going to versus the poor neutralizer is just so committed to that completely uninteresting epitope that it cannot break away. So it's supported in a permissive immune environment supports B cell diversity and low clonal competition, most of the development of neutralizing. But this case of neutralizing capabilities as compared to the individual for neutralization capacity. So again, the virus's population, B cells, it's a population, the way they're interacting with one another. And then I had a little bit of a mental health issue in that, it was the end of my postdoc. I was transitioning to a faculty position in independence, and I didn't see independence. I was working in HIV vaccine design, and I was like, I'm going to be working for someone else in the field, the larger labs that are established that do what I did, but even a more massive scale. And I was like, well, I could have just been a technician and been like a lab director. Like I wanted to be a PhD to do my own research. So once again, I applied to the FBI. So I was like, maybe this is just what I had to offer the HIV field. And they did respond to my emails this time. There was an online portal. And I was going through the hiring process, which was quite long. And the first, well, it was the longest government shutdown happened, but now the second longest government shutdown happened. So it was the end of my postdoc. I had nowhere to go. I needed a job. And a physician investigator, Dr. Colleen Kelly, was looking for a staff scientist versus moving on to an industry position. And I was like, listen, I know nothing about mucosal immunology, and I'm more of a viral evolutionary biologist who dabbled in cells. But she was willing to take a chance on me. And once again, I felt madly at this totally active research. So when we're talking about the gut mucosal ecosystem, most people think that we have the single layer of our epithelial cells separating us from the outside world. We need to absorb nutrients, we need to absorb salts and that sort of thing. Digestive tract. So we've got thin barrier separating our CD4s from the outside world. However, during receptive anal intercourse, you can have these little abrasions that let HIV gain access to those CD4s, and that's how transmission occurs via receptive killing. Sure. That's not the whole story. Your gut mucosa is an extraordinarily complex 3D immune environment. There are any number of microbial factors within your gut, short-chain fatty acids, inflammatory molecules secreted by this microbiome. The microbiome is different amongst all of us. It's interacting with the immune cells of the gut barrier. There's any number of immune cells within the gut barrier that could actually be contributing to HIV replication or restricting HIV replication. There's so much more to the story than CD4. And so one way we get at studying this or in human tissues, we can't do transmission studies in humans. But what we can do is the ex vivo challenge model. We have folks come into the clinic and they donate a couple pinch biopsies. It's more embarrassing than anything. It's not painful anymore than like you bite your cheek sometimes. We get some of these pinch biopsies, we expose them to HIV ex vivo, and then we watch how the virus behaves, we watch how the immune cells behave. And just to be clear, this is not a transmission model. This is how does the gut mucosal tissue respond to HIV and vice versa. We have some really cool things that are not CD4s. So for example, we saw a negative correlation with innate killers, so natural killer cells. And this is actually a pretty new-ish observation in the sense that up to 2016, we actually didn't think that HIV infected cells were susceptible to natural killer cell innate killing. So when you think of natural killer cells, you might think of ADAS. There are no antibodies here. These are HIV negative individuals. What's happening is that the HIV accessory protein BPU is downregulating HLA-C. So that is downregulating A and B. BPU is downregulating C. And so natural killer cells are recognizing the loss of self signal, not getting an inflammatory signal, be killing those fucking cells. Up until 2016, we thought that HLA-A and B were pulled down by HIV to avoid cytotoxic T cell responses, but C was left on the surface to prevent natural killer cells. That was a complete artifact based on the laboratory variant of HIV that was found. Turns out that in most variants, BPU downregulates C, then it's susceptible. We also see an increase in granzymes, perforin, that sort of thing in these ex vivo challenges. And again, it can't be cytotoxic T cells, it can't be ADCC, it has to be. We also saw, interestingly, the correlative positive correlation with B cells and HIV replication. So when, again, you think of B cells, you think of antibody producing cells. There are other B cells that are antigen presenting cells. And so these B cells are HLA-DR positive, and that's where we saw the positive correlation. And 500 years ago, David Montefiore also noted that something's going on with B cells and HIV. And Charles Mucosal group figured it out, which is that antigen presenting cells, they're looking out for, just stuff that's not supposed to be there. They're sticking to it and taking it in and processing it, presenting it, and it makes stew. And so they have lots of receptors on the surface that just bind to food, or in this case, C5 lectin receptors that are binding to sugars. HIV is coated in sugar. So what's happening here is that viruses don't have any kind of motility. They don't have tails. They're not swimming. They're little crystals. An antigen presenting cell comes along. The virus just happens to stick to it. The antigen presenting cell goes on to present whatever it found to the CD4. And it turns out that's HIV's cell of interest. So it's basically being carried to its target cell. What Charles Mucosal's group showed, that if you take CD4s, mix with virus on them, you really don't get a lot of replication. You have to perform lots of tricks to make CD4s produce virus. If you take that same amount of virus and mix it with APCB cells or dendritic cells, watch those cells, again mix them with those target CD4s, you get really potent HIV replication. those B cells. B cells are excellent mediators. This is called trans infection. And we think that that's actually what's happening here as well. Because again, we're talking about a three-dimensional structure. If you think about your gut, it's microns away from the outside world and all kinds of things that could do you harm. And so there's lots of antigen-specific cells there, just ready to go in action. And in this case, we think it might actually be contributing to the gut susceptibility to HIV infection. So one conclusion here is that diversity within that gut mucosal environment. So these are totally healthy people. You need just diversity in those populations. Yeah, whether you're talking about evolutionary population dynamics of African Sahara or your gut, there's differences in these cells. And that's contributing to different responses to the virus. So I'm going to bring you to some of the research we're doing today that takes that a step further. How do these interactions with the gut affect long-term health outcomes? So one thing to highlight is that every single immune cell in your body expresses estrogen receptors. That means estrogen is present, you can activate the estrogen receptor, which then acts as a transcription factor and change the transcriptome function, the number of those immune cells. And so there's lots of differences when you look at sex-based differences, it's a sex binary. When you look at T cells, B cells, natural killer cells, all those cells I just said are really important to HIV replication. Furthermore, there are so many immune relevant genes on the X chromosome. So individuals with two X chromosomes, normally one of them is epigenetically silenced, so you only get gene expression off of one of those. But on occasion, you have escape from that inactivation, you can have a higher dose of some of these immune genes in individual cells, but only if you have more than one X chromosome. Furthermore, estrogen can inhibit HIV transcription at sufficiently high concentrations. So as I mentioned, estrogen is present, it activates the estrogen receptor, which goes into the nucleus, and it turns out it just sits on the HIV transcription start site and won't let RNA fall to it. It just sits there. And so that influences latency reversing approaches, and it also influences disease progression. So again, looking at one of Eric Hunter's publications, we can see that over time, men statistically significantly over the course of infection have higher viral loads than women. Normally, a viral load, the lower viral load is associated with better health outcome. That's actually not the case. Women lose CD4s at a greater rate than men. Furthermore, women experience more of these non-AIDS comorbidities. So the development of diabetes, the development of cardiovascular disease, the development of neurocognitive issues at a greater rate than men. And some of this is from the cohorts right here in Atlanta that are just describing higher burden of these HIV, AIDS comorbidities in women, specifically young women. So, you've got a 25-year-old woman who goes into the clinic, diagnosed with HIV, but when they're 25, are you talking about cardiovascular? No, that's not a priority discussion. So, what is happening here is that I talked about how HIV influences the gut barrier using that ex vivo model. But the issue is, regardless of how you're exposed to HIV, it goes straight to the gut. It's an immune organ. It goes straight there and causes absolute chaos. And so you have lots of gut immune homeostasis. That ecosystem is totally disrupted. And then you have a loss also of the gut barrier structural integrity. That allows for translocation of microbial products from the microbiome in the systemic circuit. They're not septic. It's just a little bit of stuff that's not supposed to be there. So an individual is on effective antiviral therapy. The virus is undetectable, but this little bit of smuts getting across that damaged barrier is interacting with circulating monocytes, resulting in this ongoing chronic inflammation and immune activation that's contributing to lots of these forms of sleep. So when we think about how critical the gut is, does it make sense that the gut of a woman is functionally identical to the gut of a man? We know the estrogen in the X chromosome has such a role in autoimmunity. And I would suggest now that there should be greater CD4 activation in that deployment. And that's exactly what HIV wants to affect. There's going to be less NK7 toxicity, which I just mentioned is really important for controlling that early HIV replication. There's an increase in APC activation, which could be contributing to greater infection due to that transinfection, and an increase in inflammatory responses, which could be influencing those long-term health outcomes. So we have a study to address this quite directly, called the SING study, Sex and Inflammation, SING, where we're looking at sex hormones, sex chromosomes, behaviors, receptive intercourse. I can tell you some really fun stories on how that changes your microbiome. It just changes the microbiome, don't automatically equal dysbiosis. They're just different. Changes aren't always bad. So we're looking at the microbiome, we're looking at gut outcomes, looking at transcriptome, mucosal cytokines, tissue, HIV viral loads, that's living with HIV, and the ex vivo challenges and those that are not. And then systemic outcomes, looking at inflammatory chemokines and cytokines, that permeability markers. And this does TLR4 here, but we actually have a more broad TLR application assay. So the idea is, really to look for sex-specific patterns of inflammation to figure out more appropriate, personalized anti-inflammatory strategies, therapies for both living with HIV. We're also circling back to really what I did in my grad school days, which is looking at viral evolution and the transmitted founder, but I did my postdoc, because that damage that happens to the gut happens within days of transmission. The virus that's doing that damage is the transmitted founder virus. And so my question is, what can partially explain some of these different health outcomes in folks? Or do some of these viruses have different replicative capacity in the gut, ones that are more aggressive? Does that lead to poor health outcomes 40 years down the line versus one that does replicate upon? So we're just starting to explore that, and that's precisely what we find, that there are some viruses that just do not replicate well in the gut whatsoever, versus the range we're seeing those that do. So in this particular cohort, we're going to be able to test lots of different viruses and associate that data with long-term health outcomes in the individual. So Eric's group is already here, guys, a ton of data, inflammatory cytokines, viral loads, CD4 loss, and so forth in these same people. So we can see whether there is clinical relevance to this difference in HIV things. So, I'm going to acknowledge all the people who worked with this research, the most recent research. I have everyone here that helped with the previous research. And also just when you're doing human subjects research, 99% of the work is getting someone to walk through that door. I've always said I can teach my mom how to do these lab assays, but to get someone to come through that door from their appointment time, give you the samples, that's all the work. So I really appreciate how hard the recruitment team does to find this.
With the sex-specific differences that you were saying, is there, so you were looking at, like, when you were comparing X, Y versus X, and then also looking at sex hormones?
Yes, yeah.
Is there any evidence that people on like estrogen and hormone physical therapy are getting the same, like similar outcomes to those sex hormone or high levels of estrogen?
So there is some data with, let's say, postmenopausal women who are often been on estrogen therapy. We're beginning to generate data in transgender individuals, and some of our publications are coming out soon, but we think there's going to be a pause in funding in that direction. I'm going to work that for a bit. But so we've got some preliminary data in the lab about what happens when you add estrogen, let's say with these ex vivo experiments. And here's what I want to compare. Let's say I take natural killer cells from a cisgender man, and I ask them to kill a target cell line that lasts them. regularly in the literature, you see that it's higher in men than it is women. So there's the natural killer cells from men are better at mediating. So if a woman is menopausal, more like cisgender male. If she is on hormone replacement therapy, it goes back to. If you take cells out of a cisgender man and add nanograms per mil of estrogen, so way super physiological, you add that to the cell, that happens. So it kind of mirrors a bit what we see in our ex vivo experiments. We can add a little bit of estrogen to biopsies from human biopsies, add a lot of estrogen. And really what we see is in sufficiently high concentrations, we get that reduction in HIV replication. But we think the effect on the immune cells is during its evolution, during its mapping through the bone marrow, through its development. If estrogen is present, it's nudging that immune cell towards a pathway. And it's not the same as adding LPS to your macrophages in the next one. It's very much a biological process, which means we need to study transgender populations to really understand what's going on here in individuals who are on hormones.
That's really
She actually had the first question. And then my second question, with the viruses you found that don't replicate as well, right? If you were to add estrogen, does that things change for the virus?
We're going to have to explore that a bit more in the future. I think it could, in the sense that, in theory, but also we really need like the human population. So someone, a transgender woman, I think would be the best candidate to ask and answer that question based on what I just said. Because again, if you add too much estrogen, it does HIV just based on estrogen receptor interactions, not necessarily or broader sex specific differences. But yes, so I think what that highlights is that what I'm hoping is that really weak one, is that maybe we see a just a little higher cisgender woman that would indicate some of the things I mentioned, like a higher B cell, less with biological effects of those being.
Well, follow-up question. Back when that's a policy, one of my previous professors, he said that estrogen at extremely high level looks like testosterone. And I guess he was referring to that observation.
Well, in the sense of the way it's processed, yeah. And that's the thing, it is so complex, like sex hormones and everything is good. Because I mean, you just look at cisgender women. One of my favorite figures was like for Wikipedia that just shows the broad range in estrogen levels, even though within the same individual different cycles, it can be totally different. In our particular study, we're actually collecting biopsies during the lowest level to kind of keep it a bit more consistent. But we're missing out because some of the diversity we see might be due to differences in estrogen 2 weeks earlier. Like the best study would be to just be regularly evaluating both estrogen and testosterone. Like I really focused on estrogen because it has been directly shown to inhibit HIV. But we actually see a correlation with MK function with testosterone as well. So not only the decrease in function with estrogen, but also an increase with testosterone. And it's so hard to tease it out, right? Because one goes up, one goes down. So once again, the really transgender populations where, for example, in transgender women, in the US, the way we do therapy, we hit the estrogen up high. It's more difficult to suppress that testosterone. So we can have some folks in our study both high. So inclusive research, inclusive science is the best.
So just for clarification, in the beginning of your talk, you mentioned that there's less diversity in individual locations. Or what measure of diversity were you looking at this? Or something like Shannon or index? Because you took into account numbers of cells you were sampling at the different locations, right?
Which figure we're talking about?
Right at the beginning, when you said in different locations,
Oh,
How
yeah. So
was it?
that figure, was from, let's say, 30 years ago. And I think it was either Betty Corbett, I think it was Betty Corbett's.
Okay, she's done, I know that.
Yeah.
Yeah.
Yeah,
Another bit is you mentioned that I didn't catch this, that CD4 cell activation is higher in women. Why?
yes, so that's actually more data from Eric Hunter's paper. I didn't have time to look.
What gives rise to that? What's the?
So it's associated with estradiol, but really it is a big question as to what is everything contributing to these different outcomes. But in Eric's, that paper I referenced from
Yeah.
Eric's group, they really dig into all the different T cell types, and increased activation of the line. And that's the thing. So retroviruses, some of them require actively dividing cells to infect. HIV doesn't need it, it prefers it to personify it. So having these activated CD4s really is not a good thing in women.
And the most perplexing one is something like giving, which is HIV is sort of strange. In that you have an initial acute infection, it looks like everything's fine. And it takes a long and variable asymptomatic period. What's the current thinking on what gives rise to that long and variable asymptomatic period?
So T cells really try to do their job.
But why? Explaining that change in 10 years means you've got an almost infinitesimal change per week or month. So how can you maintain this very slight difference?
So I would say that the CD8s do a good job of controlling, and also in the sense that escape from CD8s again comes at a fitness cost. However, you've got so many latently infected cells that they can to personify things, I apologize, but they can sort of wait it out and test the waters. Say, is it safe? And until the CD8s are exhausted and they can't control things anymore, or it stumbles upon an escape mutation that it. Yes, but it is HIV grinds your immune system into the ground if you don't have antivirals.
Your talk, I had a question about, kind of the viral diversity like aspect of it. So, I guess my first question is like, have you looked into like integrated per viral community, like within actual community counts? And if so, like what kind of patterns of diversity and like the landscapes, have you conceived with that? Like, is there any kind of preferential integration of different areas and everything?
So that's Jim Mullen's research. They have mapped integration sites in lots of many cells, many participants. And so with HIV, this makes sense. Integration sites are quite variable. However, it's generally in regions that are the chromatin is open and available. So whatever regions are open naturally, then that's normally where it sits. There's not predefined integration sites. However, it's not the case where, so some retroviruses, they're doing that, and that's why they lead to cancer. So that's how we first found retroviruses in tumors. HIV doesn't do that. And so I'm not sure whether that's selective on nucleus integration sites. But also, this is also a numbers game. Every infected cell will create millions of progeny, and not all of those are going to be replication competent. So what happens is sometimes it's able to successfully reverse transcribe and plop into the cell, but actually it can't make any more function here. Or for whatever reason, they're non-functional. So there's lots of non-functional viruses are in there. What other questions?
Oh yeah, I was just curious if like, if you or anybody else has explored like, basically they like, like constructs essentially of like replication competent here. And then seeing if there's things there, in terms of like, if you have like, removal of like intraviral or saying that. One determinants like which variants will like predominate, like
So that's some Guido smart verifies. I think it's often whatever was whenever you initiated therapy, that's what pops up first. So again, I think that might be a bit of a numbers game. But yes.
I feel like this could get complicated, but have you ever looked by race? Like I imagine that there'd be different stereotypes, yeah,
So-
especially in the comorbidities.
Oh, for that, yes, so that's gonna be a non-baby, and Ego and Cecile Laurie, I get to do some of their papers, that's actually some of the stuff like this. Got there as well, could absolutely take into account, race on any of these. And so, I think it's incredibly important because, one of the most diverse sites, are cure alleles. And those are receptors on natural killer cells, yeah. So, I think that's important moving forward is to really, if you see a phenotype differences based on race, so, okay, well, what is actually contributing to that? One is the cure alleles, two is the social determinants that are contributing. I don't have the slide up here, but like, we're right now living in a pro-inflammatory environment. Stress is inflammatory. And all of that can also contribute to our health outcomes. And the groups are experiencing more stress than others. So absolutely, it's incredible. We're seeing our research helps us understand what's happening.
So for like the subpopulation of people in HIV, elite controllers, some clinical non-progressives, are they typically more men or women, or have sort of a little profile that would allow them to control that?
Yes, so that's MHC alleles. There are specific MHC alleles that are associated with elite controllers. So it's always CD8s. And it's like HLA-B57, O1 and O2. And then conversely, there's some alleles that are associated with depression. So those CD8s different, very fitness cost of those. Yes. So that was a bad, let's say 2008, where we studied these elite for us. Oh, we can make a CD8 vaccine. That's easier than trying to make a BNAV vaccine. But if you don't have that allele, you can't get any benefit. But yeah, as far as the male female, they've looked at that, and I don't know. Someone has looked at that, and I don't want to give you wrong information. But it's not. Just be the.
I have a question about treatment questions, and something's kind of bothered me for a little while, so I'm hoping that maybe you can.
There's a lot of them.
How much diversification of HIV infection take place during that infection? And I guess how good is the B cell response they have with it when it's on such a low level?
So when is latent infection? You got one cell that started, you go back from those, where you have a cell, it's infected with HIV, but it is not producing any virus. So it's just going off being a normal T cell. Like, oh, tomorrow it might start me. So that's really the question. What kicks it out? So you can see, how do we target those cells? How do you target a cell that can make a protein but is not making it? But it's just one cell. That one cell, it's called homostatic proliferation. That one cell, let's say that is a CD4 that reacts to norovirus, and you just got exposed to norovirus. And so now that one latent infected cell has now become 10,000 latent. Not a lot of diversity is being introduced there because it has the proofreading. It's being proofread by our host DNA that brings it back. It's just being more. I think it's the issue is the targets, the natural decay of infected cells versus the onset. Yeah, we can't just wait it out in the sense that we can't just give people antivirals for 40 years and then all of those infected cells just naturally pass on. So I think the diversity is there initially, it just gets multiplied.
Alright, I have one more question, is it really well that James basically deployed like to James?
I love it.
Yeah, how frequently do you see like combination or resource of those two, like for the infection setting?
All the time. So here's what happened. I did go back to the way long ago talking about HIV. But so Betty Corber and Beatrice Hahn are two investigators who, if they wanted to do the work in HIV. And they went around scooping up chimpanzee poop in acrobats with viruses and figuring out what's more similar to those variants that are affecting us now. And again, because I said HIV, the mutation rate is evolutionarily defined, we have a molecular clock. So based on all their sequencing, we hypothesized the virus positive in the map right now crossed over into humans, let's say 1890s-ish. And due to geographical isolation, evolved one way, but then let's say somebody, the copper mine, moved to a different city and took HIV with them. We knew by 1960 that HIV would have been long enough to evolve into two geographically distinct subtypes, as subtype A and subtype T. So that those initial subtypes were really just due to geographic isolation. But then, airplanes in the world. And so now what we have, instead of these geographically isolated subtypes, they're called circulating recombinant forms. So someone was infected with A and B, and so it's kind of it happens by radio. And also again, Eric has done a lot of sequencing on that dual infected individuals possibly infected. But yes, in the subtypes are ultimately going to disappear because we're no longer geographically. There's more fun stuff in that subtype C should not be, because when we do those fitness competitions, it always loses. And not even just the world, but it is a dead, when you try to compete it versus a subtype A virus, it loses 100%. And yet, subtype C virus is 50% of the world. Why? How it is not being outcompeted the way it should be outcompeted. So that's the only critical serious observation is why this less fit variant is being spread more. Is it being spread more because it is less fit? Person is living longer to spread more and so forth. But that's how it's so that's cool. Thank you.
Oh no, that's similar to some of the stuff I do. Do you have a pet hypothesis as to why? Sometimes you might be just, if not, that's cool. It's just I like those things that like exist that shouldn't exist.
Yeah.
So,
Yeah, I think it is in general because it is less fit. I mean, in the sense of, let's say, Ebola, burn out before you can get to the next person. And so, I mean, think about the most fit retrovirus on the planet is the Infectious retrovirus. We're all infected with Infectious because there's no detriment to have it. So maybe that's leading seeds or things. They can be quiet and poison. So 2% of virus against certain teachers.
Great, yeah, thank you.